becomes very stiff. There is bronchial smooth muscle changes. Bronchial smooth muscles, both there is hypertrophy and hyperplasia of the bronchial smooth muscles. They increase in number, and bronchial vascular changes. That means there is edema. There is edema of the bronchial vascular wall, and that causes narrowing of the lumen. So there are many changes apart from these smooth muscle changes in asthma. And we know that in asthma, a local trigger can produce a generalized bronchoconstriction. Now, my PGT friends are here. We all know that what is the predominant nerve supply in the airway epithelium? Is it sympathetic or parasympathetic? It is usually parasympathetic. Okay, parasympathetic and cholinergic. The predominant constrictor response in airway is predominant and parasympathetic. This is important because we use various kind of bronchodilators to treat asthma. This is either beta-2 agonist or anticholinergic. But the predominant constrictor response in the airway is cholinergic and parasympathetic. And the predominant relaxant effect on the airway is again parasympathetic, non-cholinergic. So sympathetic has a very negligible contribution in the treatment of in the whole airway, though we use these beta-2 agonists very frequently in treatment of asthma that has different causes actually. So whenever there is a predominant contractile innervation of the airway smooth muscle is parasympathetic, cholinergic. So what happens in neurogenic inflammation, and this is brought about by the tachykinins, what happens? We have nerve endings beneath the epithelial cells. There is epithelial shedding. So nerve endings are now exposed. Now what happens in the neurogenic inflammation? There is excitatory tachykinin secretion. These excitatory tachykinins are neurokinin A, and that acts on neurokinin 1 receptor and neurokinin 2 receptors, and there are substance P. So that produces generalized bronchoconstriction. And we have also inhibitory tachykinins. Those are vasodilatory, and they are mainly the vasoactive intestinal peptide and the nitric oxide. Okay, so these things are required for the research purpose actually, but we have to know for our PGT friends, these are very important for the examination purpose actually. How a local inflammation becomes a systemic inflammation, this is very important. Now, we know in asthma there is airway hyperresponsiveness. That means airway constricts to any kind of stimuli. Why it happens? This is due to the parasympathetic stimulation. But parasympathetic stimulation directly doesn't cause this bronchoconstriction. It causes through multiple pathways. There is parasympathetic stimulation that stimulates the submucosal glands because all these things are supplied by the parasympathetic nerves, and there is stimulation of the inflammatory cells that causes release of the inflammatory mediators, and that causes recruitment of the leukocytes and activation of the leukocytes. So parasympathetic stimulation causes inflammation, recruitment of leukocytes. These leukocytes release various neurotransmitters, and there is bronchoconstriction and airway hyperresponsiveness. What are these bronchial constrictor agents? There are autocoids, that means serotonin, histamine. There are proteases. There are heparan sulfate. So various things are there which causes bronchoconstriction. So it is a response to the inflammation which is going on into the airway. That is the cause of the airway hyperresponsiveness. Now we now know that asthma is not a single disease. Even some papers have said that asthma is like a fever. In fever we have different causes. Asthma is a heterogeneous disease having various phenotypes and various endotypes. And this is a constellation of various things which is under a single umbrella we call asthma. But I think this, in near future we are going to call asthma by different names. We either will call it as a T2 high asthma or it's a T2 low asthma, because the treatment will change according to that. And all these things are important to know, because it will when in the next portion will come about how the air pollution causes different shift in this inflammation, then whether it causes a T2 high asthma or T2 low asthma, this concept is very important. So asthma is basically now divided by the molecular immunology method into T2 high or T2 low. What does it mean by T2? So we know our immunogenic response is basically of three types. It may be a T1 type, it may be a T2 type, or it may be a T3 type. I will come one by one what are those things, because if you don't know what are those things, we cannot go forward actually. So basically asthma is T2 high or T2 low. T2 low means T1 high, T3 high or polygranulocyte. Now what happens when an antigen just comes inside the body? What happens? The antigen presenting cells engulf the antigen, and after engulfing the antigen, it presents the antigen to a T cell, and these T cells are usually the naive T cell, which are further differentiated into helper T cells. So antigen is engulfed by the antigen presenting cells that is presented to a naive T cell, and this naive T cell further differentiate into various type of helper T cells. Now there are various helper T cell subsets. It may be a Th1 cells, or that Th1 cells is required for intracellular killing of bacteria. So when the bacteria or viruses are intracellular, we need this T1 cells pathway to kill that bacteria. Now Th2 cells, what they do? They are part of the humoral immunity. That means they produce antibodies and they kill the extracellular pathogen. So for intracellular pathogen killing, we need Th1 cells. For extracellular pathogen killing, we need Th2 cells. What is Th17? This is a part of T3 immunity where it kills fungi and extracellular bacteria. So for different types of organisms or different types of inserts, we have different types of immune reactions going on inside our body. For intracellular, we have Th1. For extracellular, we have Th2, and for some specialized extracellular, we have Th17 type of immunity. And we have T regulatory cells. What are the T regulatory cells? So these are actually suppresses the T cell response. When an inflammation is going on, it has to be suppressed at some point of time. Otherwise, it will be excessive. There will be autoimmunity. So T regulatory cells are actually a subset of cells which suppresses the immunity and prevents this autoimmunity, and we have follicular T cells that produce antibodies. So I think it is getting complicated, isn't it? Yes. So we have two types of immunity in the body. We have adaptive immunity and we have acquired immunity. Like we have BSF in the border and we have military. Okay. So whenever there is an invasion, foreign invasion, what happens? First the BSF attacks. So BSF is the innate immunity. They are not antigen specific. They will attack all the foreign things. But if they cannot control the invasion, they will inform the military, and they will come and they will reinforce the attack. So this military is the part of the acquired immunity. Now what type of attack they will develop, that depends on the pathogen. Whether the invasion has occurred from Rajasthan or whether the invasion has occurred from Kashmir. If the invasion has occurred from the Rajasthan, one type of immunity will play, or one type of soldiers will be deployed there. If the invasion has occurred from Kashmir, different types of soldiers will be deployed there. Those who are specialists in those regions. So if there is intracellular organism, some different types of immunity will come into play. That is the Th1 immunity. If the extracellular organisms, where the antibody has to be produced, there is Th2 cells. In COVID, what happened? What type of immunity played a role? Both the Th2 which produced antibody against COVID and intracellular Th1 cells also played a role, where there is memory cell formation, and that actually caused a long-term protective effect in humans. So in COVID it is said that Th1 immunity is is the desired one than the Th2 one, because if you have a Th1 immunity, it will give a long-term protection. So Th1 produces memory cells and Th2 also produces memory cells, but Th1 memory cells persist longer in human body. And this all inflammation has to be suppressed at any point of time. That's why the T regulatory cells are important. They will suppress the inflammation at some point of time. So they are the controller of inflammation in the human body. Now, what I am telling about, what happens? An antigen presenting cell, entered into the human body. Now, antigen presenting cells are there, like the dendritic cells are there. They engulf the antigen, process the antigen, and in presence of interleukin 12, they are differentiated into this Th1 helper CD4 T cell type. Okay? And now they secrete the interleukin 2, and that promotes the growth and differentiation of other T cells, or IFN gamma that activates the macrophages and that invites further cells, and the immunity and the reaction continues. So T1 immunity for intracellular pathogen, and this is brought about by the interleukin 12. And for T1 immunity, the signature is IFN gamma. So if you want to measure the T1 immunity of any organism, you have to measure the IFN gamma or interferon gamma for that. What is the level of that IFN gamma in that person? For T2 immunity, what happens? A similar thing. The antigen presenting cell is there. It engulfs the antigen, but now different interleukin comes into play. This is interleukin 4. Previously it was interleukin 12. And that differentiates these naive CD4 T cells into Th2 types of cells, which secrete 3 main type of interleukins, and they are the hallmark of the T2 type of immunity. Now, previously it was called Th2 type, but now it is called T2 type because apart from the CD4 T cells, there are other cells also which comes into play, like the innate lymphoid cells. There is significance of that, I will tell you later. So what happens? These Th2 cells secrete the interleukin 4. Interleukin 4 that stimulates the B cells to produce Ig antibodies. Interleukin 5 that activate mast cells and eosinophils, and interleukin 13. This is very important interleukin in asthma, and that produces mucin production, fibrogenesis, bronchial smooth muscle dysfunction. Various effects are caused by this interleukin 13. And as we all know that now there are various types of monoclonal antibody directed against interleukin 4, 5, 13, and in our city we have antibodies against interleukin 5. So these 3 interleukins are a hallmark of the T2 inflammation in asthma. Now what happens in T3 immunity? The same process. Antigen presenting cells engulf antigen. Now here is an interleukin 6 and TGF beta, and in presence of that it is differentiated into Th17 type of cells which secrete IL-17 or interleukin 12. IL-17 recruits neutrophils at the site of infection, and interleukin 12 stimulates epithelial cells to produce antimicrobial peptides. So we saw that different types of interleukin actually causes different type of shifting of this Th helper 2 cells into different group. Interleukin 12 causes shifting towards Th1 type. Interleukin 4 causes shifting towards Th2 type, and interleukin 6 causes shifting towards Th17 type, and that is being determined by the nature of the antigen or immune susceptibility of that particular host. Now what does T regulatory cell does? It is a potent suppressor of the macrophage function, and interleukin 10 is said to be an anti-inflammatory interleukin, and it induces the interleukin 10 and TGF beta. So for T2 high asthma, we require, or the signature of the T2 high asthma is interleukin 4, 5 and 13, these 3 interleukins, and that causes eosinophil recruitment into the airway, and the cells involved in T2 high asthma are CD4 T cells, group 2 innate lymphoid cells. These cells are very important and recently been described. Previously we thought that all this T2 type of inflammation is by the CD4 Th2 type of cells. And these CD4 Th2 types of cells are very much responsive to steroid. But we all have seen some patients of asthma does not respond even to high dose of steroid. Why that? Because in those patients, this interleukin 5 secretion or interleukin 13 secretion are not by the CD4 Th cells. They are by the group 2 innate lymphoid cells. And these innate lymphoid cells are not responsive to steroid like the CD4 T cells. Because after the discovery of this group 2 innate lymphoid cells, now we call it as T2 high asthma or T2 low asthma. Previously it was Th1 high asthma, Th2 asthma, because Th is no longer is being used, because apart from the Th, these innate lymphoid cells has similar action which are not responsive to steroid. And the effector cells in T2 high asthma are the mast cell and the basophils, and they mediated the Ig mediated changes in asthma. So Ig antibody is formed. They are deposited in the when there is a exposure to the antigen, they attach to the antigen, and there is high affinity Ig receptors on the mast cell and basophil. They attach there, and there is degranulation of the mast cell and basophil, and release of various kind of bronchoconstrictor mediators. So this is the picture taken from the latest edition of the Marinadal, where you have the various kinds of allergen. It may be a microbiome, or air pollution, or unknown chemicals. When they are present, this is the airway epithelium. All this antigen has a specific part which is recognized by the pattern recognition receptors in the epithelial cells and the TLR receptor, that is toll-like receptor. So they know the antigen, they attach with the antigen and secrete this kind of cytokines like the thymic stromal lymphopoietin, IL-33, GM-CSF, TGF. Just remember these four cytokines, because they are very much important when I talk about how air pollution causes asthma. So the first insert, this antigen is recognized by these receptors, and they secrete this kind of cytokines, and these cytokines actually invite the dendritic cells, and the dendritic cells then present the antigen and process the antigen and present it to CD4 T cells, and either there is a Th1 response, Th2 response, or Th3 response depending on the interleukin secreted there. So the initial response is by secretion of this thymic stromal lymphopoietin or IL-33 or GM-CSF or TGF. You know all the full form of these things. And this is a part of the innate immunity, and this is a part of the adaptive immunity. Now the T2, what is the incidence of T2 low asthma in asthmatic patients? For less severe asthma, around 60 to 70% of asthmatic having a T2 low asthma, and for severe asthma, around 50% of the asthmatic is having T2 low asthma. Now we know many things about T2 high asthma, but for T2 low asthma, the researches are not very widely available, and this part of the asthma is not being discovered so well. T2 low asthma doesn't respond to steroid very well, and we need some other treatment. Biologics have to be used for many of them. Now, what is T1 high? T1 means intracellular pathogen. If you remember, T1 intracellular, interleukin 12. Okay? So that protects against intracellular microbes. It is mediated, the signature is interferon gamma, and the cells involved, group 1 in innate lymphoid cells. In T2 you have group 2, and in T1 you have group 1, there are CD8 cytotoxic T cell and CD4 Th1 cells. In T3 high, that protects against extracellular bacteria and fungi. The interleukins involved, interleukin 17 and interleukin 22, and activation of the mononuclear phagocytes. And that was recruits neutrophil and group 3 innate lymphoid cells, Th17 producing cells are the final cells. Those will be recorded in this T3 type of immunity. Now apart from that, we divide asthma according to the cellular phenotype, whether it's a eosinophilic asthma, whether it's a neutrophilic asthma, whether it's a posse granulocytic asthma, that means no cells are there, or it's a mixed cell, both eosinophilic and neutrophilic. So cellular phenotypes, that is done by the sputum induction, either neutrophilic, eosinophilic, neutrophilic, mixed and possibly granulocytic. Eosinophilic, if you have more than 2 to 3% of the eosinophil in induced sputum, that is called eosinophilic phenotype of asthma. And it is a classic atopic asthma. What is meaning of atopy? If you have a very high Ig, that is called atopy. So eosinophilic asthma is a classic atopic asthma. Neutrophilic asthma, if you have a sputum neutrophil count more than 60 to 70% in the induced sputum, we call it a neutrophilic asthma. Normal neutrophil count in the sputum is less than 50%. And this kind of neutrophilic asthma occurs due to repeated infection, that means the viral infection or bacterial infection cause neutrophilic asthma, obesity can cause neutrophilic asthma, smoking and pollutants can cause neutrophilic asthma. But in our presentation we will see air pollution can cause both neutrophilic and eosinophilic asthma. Mixed means there are both neutrophils and eosinophils, and these are reported in refractory cases of asthma. And post-granulocytic means in less severe asthma we do not have any particular kind of cells in the airway epithelium in the induced sputum. Now, after phenotype, we now divide asthma according to the endotypes. What are the meaning of endotypes? Endotypes means either it's a T2 high endotype or T2 low endotype. So, what is the meaning of phenotype? Phenotype meaning the host gene interaction actually produces various kinds of external manifestation. That is the phenotype. And what is endotype? The biological pathway leading to this different phenotype is called the endotype. So asthma is now divided into T2 high endotype or T2 low endotype. T2 high endotype, what is the characteristics? They have a very airway hyper responsiveness. That means they have bronchoconstriction on trivial stimuli. Exposure to trivial stimuli, they have very high Ig, high eosinophil in the bronchoalveolar lavage fluid or the blood, more subepithelial fibrosis, that means they have more chance of airway remodeling, higher intraepithelial mast cells, and more responsive to steroid. This is the T2 high endotype characteristic. And what is the characteristic of T2 low endotype? It may be either T1 or T3 or systemic inflammation can cause asthma. What happens in obesity? In obesity, there is increased interleukin-6 secretions. Interleukin-6 is produced by interleukin-1 beta. So that causes secretion of TSLP, that means thymic stromal lymphopoietin, interleukin-33, and the cascade starts from the systemic inflammation. So here the inflammation is not local. Local inflammation is a trigger, but the systemic inflammation is the driver in obesity. Or there may be some other non-inflammatory mechanism like airway smooth muscle dysfunction may cause asthma. Epithelial barrier dysfunction. If the first defense is defective, the epithelial barrier is defective, then that can cause asthma. Or there is neuronal abnormality, the neurokinin mediated inflammation will go haywire. Or if there is mucin dysfunction, the mucin is produced in the airway to protect against this microbe. If there is excessive mucin, then that will cause an airway bronchoconstriction. Now, air pollutants. Coming from the asthma, now we are entering what are the air pollutants. We all know there are different types of ambient air pollutants. There are respiratory pollutants which causes problem in the respiratory system. What are they? And there are non-respiratory pollutants which enter through the respiratory route but causes problem elsewhere. Or there are carcinogens which produces cancers. What are the respiratory pollutants we are concerned about in World Asthma Day? Particulate matter, sulfur dioxide, nitrogen dioxide, and ozone. And for non-respiratory pollutants which enter through the lung but causes problem elsewhere are carbon monoxide, lead, benzene, and cadmium. And for carcinogen, we have polycyclic aromatic hydrocarbons, we have N-nitrosyl compounds, asbestos. There are a lot of there, but these are important. Now ozone, I have told you, ozone is not a natural constituent of air. We have ozone in two layers. We have stratospheric ozone, we have tropospheric ozone. Stratospheric ozone protects us from the UV rays, but tropospheric ozone or the ground level ozone actually causes various types of ill health. So what happens? It is produced by the oxides of nitrogen and volatile organic acids. in presence of sunlight, it produces this O3 or ozone. Now, the gas and the particulate matter, there are air pollutants as a mixture of gas and particulate matter. Okay? So, SO2 is a gas, ozone is a gas, nitrogen dioxide is a gas, and the particulate matter, PM 2.5, PM 10, PM 0.1, they are the particles. So, gas has different behavior and the particulate matter has different behavior. And the property of the gas actually determines how much gas will be deposited in the lungs. Either it may be a soluble gas or insoluble gas. If a gas is soluble, like sulfur dioxide, it will be absorbed in the upper part of the respiratory tract. It will not go downward. And the particulate matter deposition depends on the size, aerodynamic diameter, and the composition of the particulate matter. The coarse particle, which have a diameter of more than 10 micron or PM10, actually are filtered by the nasal ciliary apparatus, or they are impacted in the airway because our airway is tortuous. The fine particles actually either sediments in the lung because of the gravity, or they go to the alveoli and deposited in the blood vessels. And ultrafine particles are very unstable. They undergo chemical reaction and agglomeration, and they are very much unstable particles. So we are concerned about these fine particles because they produce local effects and the systemic effects both. Now the deposition of these particles depends on various things, depends on the flow rate of the flow rate of breathing, depends on the host defense. If you are exercising, then your respiratory rate is high, your tidal volume is high, your inspiratory flow rate is high, your respiratory flow rate is high. That means you will have more deposition of the pollutants into your lung. So that's why we always tell, do not exercise too much during winter season where the air pollution levels are very high. And host defense, we have the cough reflex as a defense. Cough actually expels the amount of allergens that is deposited in the airway. Mucociliary escalator, that means the ciliary beat, and alveolar macrophages. These are the host defense which actually causes or actually protects us against various of these pollutants. So actually this is the balance between the deposition of the particle and clearance of the particle that actually determines the disease onset. If the deposition is more than the clearance, then the patient will have starting symptoms of air pollutant related diseases. Now, how these different pollutants cause different manifestations? Suppose we have the robust data about the particulate matter. Particulate matter causes various types of lung damage. What are the mechanisms by which it causes? The particulate matters have surface particles like iron and quinones, and they are strong. They have strong electrical charges, and they form the reactive oxygen species like hydrogen peroxide or hydroxyl radical. We know we have an oxidative system in the body and antioxidative system. Any disease is some diseases are produced by the oxidative stress, like any chronic inflammatory disease, and this is being counteracted by the antioxidant system. So if you have a problem with the antioxidant system, the oxidative damage will be more. So these are the all oxidant radicals, the reactive oxygen species H2O2, OH, and these are produced by the particulate matter and they are deposited in the lung, and these increases the oxidative stress in the lung. The particulate matter also carries the endotoxins, and this produces or triggers inflammation, and this type of endotoxin actually produces Th1 type of inflammation, and there is antigen presenting cell mediated inflammatory response, and there is Th2 and Th17 upregulation. So that's why particulate matter, if it causes Th2 upregulation, it will cause T2 high asthma. If there is Th17 upregulation, it will cause T2 low asthma. So it can produce both. Particulate matter can produce both T2 high and T2 low form of asthma. And it also causes Th1 downregulation, and there is increased autophagy and apoptosis of the lung epithelial cells. What is apoptosis? Programmed cell death. And what is autophagy? That is also
normal cell death, মানে fusion of the cell with lysosomes.